Hello, everyone. Thank you for joining the Microbial Insights monthly webinar series. Today, I am delighted to share with you a presentation on 25 years of BioTrap samplers. I know many of you are familiar with this tool, and so hopefully you're going to learn some new information today and also maybe see some things that are familiar to you. But we, as always, really appreciate that you're taking the time to join us on this series. And it's an absolute pleasure for me to be able to introduce our guest speaker today, who is a dear friend and colleague that I've worked with for over 20 years, Dr. Carrie Sublette. Dr. Sublette recently retired as an emeritus professor of chemical engineering and geosciences and a Sarskis professor of environmental engineering at the University of Tulsa. He has over 40 years of experience in a wide variety of bioengineering and soil remediation problems, including bioremediation of hydrocarbons, chlorinated hydrocarbons, and fuel oxygenates in soil and groundwater. He's done a ton of forensic analysis and used molecular tools from the very beginning. So Dr. Sublette is a wealth of knowledge and a personal resource and mentor for me over the years. He has 13 patents, over 120 publications. He was the chair of the annual IPEC conference for 26 years. And was a director of the International Petroleum Environmental Consortium, which was an EPA research center focused on oil and gas environmental issues. So we have an incredible expert here for you guys who's going to do this presentation, and at the end we'll have Carrie answer some questions for you guys. So before I let Carrie take it away, I just want to remind you that, as always, the webinar is being recorded. It'll be available on the resource section of the Microbial Insights website by the end of this week. We will be sending out an email to everyone with a link and certificate of attendance following the webinar for those people that registered. There are also some additional handouts that you can see about biotraps that are in the GoToWebinar panel that you can download, and you can certainly reach out to our team at info@microbe.com if you have other questions. And then, as I mentioned, when Carrie is finished speaking, we'll go through and answer your questions, as many as we have time for after the presentation. So please welcome us in or join us in welcoming a true environmental expert to our panel today, Dr. Carrie Sublette.
Well, thank you, Dora, for that introduction. I appreciate it, and I'm happy to have this opportunity to talk about a favorite topic of mine, and that's biotraps, which I've been using now for over 20 plus years. And before I begin, you're going to see that some of the slides in my presentation are going to go by pretty quickly today to make a small point, and then we'll go on. And I apologize for that. That's just me trying to get as much information as possible into this webinar. But I invite you to either contact Microbial Insights or contact me directly with the email that you see on the screen now, and you'll also see it on the last slide if you would like a copy of this presentation. And I'd also like to invite you that if you have any questions about this presentation that we don't have time to answer at the end of the webinar today, please feel free to send me an email, and I will do my best to answer your questions. Now, as Dora said, I know that some of you out there are familiar with biotraps and some are not. So I'm going to kind of begin at the beginning here and ask ourselves, what is a biotrap sampler? A biotrap sampler is a passive sampling tool for microorganisms, but it's really more than that, because the important thing about a biotrap is what it collects. As indicated here, a biotrap collects environmentally competent active microorganisms in the environment that you're sampling. And the reason for that is that microorganisms have to get on the inside of our collection medium that you see here identified as bioset bees. They have to get on the inside of those bees and grow and reproduce there under in situ conditions to actually be detected. So a biotrap, I think one of the most important aspects of a biotrap is that it allows you to focus on those organisms that are having the greatest effect on the microbial ecology, and those are going to be the ones that are actively growing and reproducing and carrying out biotransformations in that environment. You can kind of think of that as separating these organisms from all the microbial noise that may exist in that medium. In any medium, whether it's groundwater, soil, whatever, there are going to be a lot of microorganisms that are there, but they're really not doing anything right now because the environmental conditions are not right, the nutritional conditions are not right. So the biotrap allows us to focus on those organisms that are actively growing and reproducing in that environment. Another advantage of a biotrap is that it provides an integrative sample, because these biotraps are typically incubated for 30 to 45 days, usually in groundwater, but it can be soil, sediment, etc. And during that time, the biofilms on the inside of the bioset beads are coming into equilibrium with that environment. So you've got a sample that's basically integrated over the time of incubation, as opposed to going in and taking a single sample that gives you kind of a snapshot. Now, once the biotraps have been retrieved, they can be analyzed, as you're going to see in some of the examples here, by a variety of molecular biological tools, analytical chemistry tools, and also, as you'll see too, we can also use stable isotope analysis to provide information for us. Excuse me just a moment, I forgot to turn off my phone. There, sorry about that, guys. So how do biotraps work? Well, the magic of biotraps is in the collection medium, and that's what we call bioset beads. If you're not familiar with this material, these beads are about 3 to 4 millimeters in diameter. They're made of only two materials, Nomex and powdered activated carbon. Nomex is the same material that's used in fire retardant suits, so it's very thermally stable, and you'll see why that's important in a moment. So you can basically think of Nomex as kind of holding powdered activated carbon in a three-dimensional matrix. So it has a very, very high porosity, has a very high surface area, as you can see here. And because of the thermal stability of Nomex, these beads can be heat sterilized at 270 degrees centigrade before they come to you. And that should be important to you, because that means that they can be thoroughly cleaned of fossil biomarkers, so that you know that not only are they sterile when they come to you, but they're clean of these fossil biomarkers that might confuse the picture if doing biomarker analysis. And as I said earlier, they're very rapidly colonized by active microorganisms. In the photographs you see here in the upper right-hand corner, that's a cross-section of a bioset bead. The second photograph is a biofilm on the inside of a bioset bead. And the third is also another scanning electron micrograph, but this one at a very high magnification, showing a microorganism on the inside of a bioset bead. And as you can see, it's in the process of cell division. And you can see those little strings there, which is dehydrated EPS, which is the material that basically holds microorganisms together in biofilms, or in this case, holding on to the solid surface of the interior of the bead. So what can I do with the biotrap? Well, there are a number of things we can do. Probably most commonly are these 5 here. First of all, we can determine if a degrader of a contaminant of concern is present and active in the environment. We can make predictions as to the response of a microbial community to a potential remediation amendment that we might wish to try to use for biostimulation. We can evaluate the potential effectiveness of a bioaugmentation culture. And in situ, in the field, we can actually evaluate the in situ response to bioremediation or bioaugmentation to make sure that the potential of that stimulation or augmentation is being realized in the field. And lastly, as I'll show you in a special application, we can provide irrefutable proof of the biodegradation potential of certain target compounds. So what does a biotrap look like? This is probably the most common type of biotrap that you would see This is the typical biotrap for just doing standard sampling of groundwater, even though it's also used in sediments. It's also been used in soils, etc. You can get an idea of the size from the penny that you see there. They're about 3 inches long and they hold about 250 bioset beads. How do we deploy bioset samplers? Typically, as I said, the typical application is in groundwater. But again, I'm going to be speaking a lot about groundwater, but I don't want to leave you with the impression that that's the only medium that can be sampled. It's the most common medium that we're sampling with biotraps. But if we're sampling a groundwater monitoring well, we would purge that monitoring well, and we would suspend the biotrap from the casing, the top of the casing, and we'd suspend that biotrap at whatever depth interval we might be interested in. And if you have an aquifer, an unconfined aquifer that has a lot of fluctuations in the water level, and those are anticipated during the time of incubation, we can actually suspend that biotrap from a float, so you can maintain it at a given depth no matter what happens to the groundwater level. So where do we deploy biotraps? Again, we're looking at contaminated groundwater here, just a little representation of a map of a site with contaminated groundwater and the source area identified there. Typically, what we would recommend is to deploy biotraps along the axis of that plume, certainly a biotrap in the source area, down gradient of the source area, and also recommend a background well up gradient of the plume. Those would be our primary locations. Our secondary locations, we might also sample down gradient. We might also sample the periphery of the plume to find out what's going on in those areas as well. So I want to address a few questions that I've gotten over the years with a couple of slides that I'll run by very quickly. One question that I used to get a lot was, well, how do microorganisms get on the inside of bioset beads? Take, for an example, an organism like the Helicocoides that we're all very interested in. It has no known mode of mobility. It doesn't have a flagellum, doesn't have any twitching mobility or anything like that. So how does the Helicocoides get on the inside of the bead? And basically the answer is Brownian motion. Takes basically results in the microorganism coming in contact with the bead, and the environment in that bead is now basically enriched in nutrients because of the active surface of the powdered activated carbon on the inside of the bead, and microorganisms then grow into the bead if they're environmentally competent under those conditions. So that's the way that we get the Helicocoides in the inside the bead. What you're looking at here is just a little histogram taken from the Microbial Insights database, specifically of a number of sites where we knew active degradation of or reductive dechlorination of halogenated hydrocarbons was going on, and just showing you some of the numbers here that we're seeing in terms of concentrations of the Helicocoides in the beads, and obviously they're quite high in those types of situations. So the Helicocoides is having no trouble at all getting on the inside of the bead. Now, if we also dig deeper into the Microbial Insights database and look at biotraps versus groundwater samples, that's another question I've gotten in the past. How does a biotrap sample compare to a water sample? And here I'm looking at the Helicocoides, and again, looking at now a larger subset of the Microbial Insights database, which contains tens of thousands of sets of data. And what I want you to see is that at a log of cells per unit or cells per bead of greater than 1, if you look in this database, you see that as far as the water samples are concerned, the biotrap samples are concerned, the distribution is basically the same. In fact, above a log of 1, the means are equivalent statistically at confidence levels of greater than 95%. However, things are different at at concentrations below log of 1. And why is that? We believe that the reason for that, as you can see here, you in the 0 to 1 range, you're seeing higher concentrations in water than you are in biotraps. And why is that? And that's because at these very low concentrations, the microorganisms are not rapidly growing and reproducing. In fact, they're rather stagnant. Therefore, they show up in water samples, but they don't show up as well in biotrap samples because there's lesser growth going on. So when it comes to an organism like the helicocoides, if your question is, are there low concentrations of the helicocoides that are currently inactive, but might be potentially stimulated, I would use a water sample to try to find that out. If your question, though, is what is the concentration of actively growing the helicocoides, say in a site that you're trying to stimulate, then I would use biotraps. Similar results here for a hydrocarbon impacted site. And as you can see, a wide distribution in the MI database, but obviously hydrocarbon degraders have no problems at all getting on the inside and actively growing, reproducing in the bioset beads. One curiosity here, notice that some 14% of the samples that were used in this particular analysis was not detecting hydrocarbon degraders using biotraps. Well, we've all been told that hydrocarbon degraders are ubiquitous, so why is that? And the reason for that is that there are a lot of environments where hydrocarbon degraders may be present, but they're not actively growing and reproducing. And if they're not actively growing and reproducing, the biotraps won't see them. So again, with the biotraps, we're focusing on those organisms that are rapidly growing and reproducing and are responsible for the biotransformations that are going on in that environment. And I could show you many plots like this, but I could tell you that so far we have seen no evidence of any bacteria or archaea that will not readily grow in biotraps. We haven't seen anything in water samples that we haven't also seen in biotrap samples as well. So very quickly, I want to make sure that you understand that what you saw was a standard biotrap for standard groundwater sampling, but we can design a biotrap for any application that you may have. For example, here is a flow-through biotrap that's being used in a drinking water distribution system. Here's some custom-made biotraps that are sampling an aqueous layer below a kerosene solvent and a solvent extraction system. Here are some biotraps that are attached to a coupon holder for sampling organisms for mic in an Alaskan pipeline. Here's a biotrap specially designed for a client that wanted to look at different partial pressures of oxygen in the subsurface and the effects on rates of degradation of hydrocarbons. So here we have these gas-fed biotraps. You can see the pictures and the diagram there. Here's another custom design for sampling and sediment on the ocean floor. These were actually taken down by divers and driven into the sediment on the ocean floor, looking for degradation potential of hexane, as I recall. So there are lots of different biotraps take lots of different forms, and that's a message that I want to leave you with. Any kind of container that allows bioset beads to come in contact with the medium that is being sampled, that is a biotrap. And we can design a biotrap to get into any kind of environment that you would be interested in sampling. So let's go back to those bullet points we saw earlier about what can we do with a biotrap. And the first bullet point was determine if known degraders of a contaminant of concern are present and active. So let me give you some examples here. This is just a site where PCE is undergoing a reductive dechlorination, and we've just got 2 biotraps in the plume area. And what you can see here is we have the helicocoides concentrations of about 10 to the third. We also are picking up vinyl chloride reductase, which, if you're not familiar with that, is one of the enzymes that's involved in the last step of the reductive dechlorination process. So it's frequently a functional enzyme that we're looking for. And also marked on here is the level of 10 to the fourth, which is generally accepted and 10 to the fourth cells per bead, which is roughly equivalent to 10 to the fourth cells per milliliter of actively growing dihelicocoides. And that's generally accepted as the level that's needed for meaningful rates of degradation of chlorinated hydrocarbons. So if I was looking at data like this from a site and I wanted to know, ask myself questions like, well, is reductive dechlorination possible? Well, certainly it is. You've already got a pretty healthy concentration of active dihelicocoides, but we're not quite at those levels that are required to get those rates to the point to be useful in the remediation of that site. So then we would look at data like this and we would consider then adding an electron donor to stimulate the growth of dihelicocoides. And then, as I said earlier, we can also use biotraps then to determine or demonstrate the effect of that electron donor or that amendment in the site after we've injected it. And I'll show you some examples of that. I've lumped a couple of these others together for reasons that I think will become obvious in a moment, but we can predict the response of a microbial community to a remediation amendment biostimulation. We can also evaluate the potential effectiveness of a bioaugmentation culture. And in doing so, basically what we do is use a biotrap and another device that you might call this a second step in the evolution of biotraps. To use a biotrap and a device called an in situ microcosm, which you see on the left-hand side of the screen here. Now, the in situ microcosm contains a biotrap for sampling the microbiology, but it also contains passive diffusive samplers for organic material to measure contaminant concentration, daughter product formation, etc. Again, the biotrap, we can use qPCR. We can use a technique called stable isotope probing that I'll be telling you about to quantify the specific microbial populations and the processes that are going on. We can also have a passive diffusive sampler for the inorganics so that we can look at redox conditions, changes in electron acceptors, etc. And then we have this device here called a sampler. Now, this is just represented by a sponge here. There are different kinds of devices that can be used to supply material. The material we're supplying here is a biostimulation amendment of some sort. And as you'll see, they're depending upon the contaminants that we're working with, we might use different amendments here. But that's basically what the inside of an in situ microcosm looks like. Now, these are going to be temporarily, these are going to be primarily deployed in sets. And what I've shown you here are three in situ microcosms. But we might also deploy these in pairs. But in these sets, there's going to be one that does not contain any sort of remediation amendment, and that's going to be our control, or sometimes we refer to as our MNA or monitored natural attenuation control. And then we can also attach additional in situ microcosms in which we might introduce a remediation amendment. And what we've shown here is the potential for testing two remediation amendments that we've labeled treatment option 1 and treatment option 2. Notice that these in situ microcosm units are separated from each other by baffles so that there's no crosstalk between the different units. So just to give you an example here, what might we be looking at for a hydrocarbon impacted site? Perhaps we would have 3 units, one being our control with no remediation amendment. And here in this example, we have And one treatment option, which is using an oxygen release compound. So basically we're making oxygen available, or we might have another treatment option that's shown here, the BioStim, where we might be providing sulfate, for example, as a terminal electron acceptor. So the objective here would be to compare the effects of aeration or making oxygen available from an oxygen release compound to using sulfate as a terminal electron acceptor and comparing that to the absence of those remediation amendments. So when we, after incubating these, typical incubation 30 to 45 days, for example, if we focus on the biotraps inside the in situ microcosm, if we analyze the control biotrap, that's basically telling you what the microbial population looks like without any remediation amendment, what the active component of that microbial population looks like. The second unit here is going to show you how that microbial population is going to change if you make oxygen available. Is that going to shift in the right direction? Are you going to stimulate the aerobic organisms that are necessary for hydrocarbon degradation or capable of carrying out hydrocarbon degradation with oxygen as the electron acceptor? And the last unit would answer the same question about sulfate. You know, what's going to happen if sulfate is supplied to the microorganisms in this particular aquifer, and how does that compare to the absence of sulfate? So in other words, is sulfate going to stimulate the growth of hydrocarbon degrading bacteria? If I look at the same kind of idea now using, looking at chlorinated hydrocarbon impacted sites, again, one possibility, again, I keep showing these in threes, but remember, we don't necessarily have to have three. We can be, if we only have a question about one amendment, we would use two. But for a chlorinated hydrocarbon site, say we might have three of the in situ microcosms. One again would be a control. There would be no biostimulation amendment, no remediation amendment, no bioaugmentation. The second unit might be what we call a biostimulation unit, and this one would have some sort of electron donor supplied. And in the last unit, we've labeled here BioAug. The BioAug might be including a bioaugmentation culture as well as an electron donor. And how do we supply that bioaugmentation culture? We will incubate, in the laboratory, we will grow the bioaugmentation culture in the presence of biocept beads. They then grow in the beads, and and basically load the beads with that bioaugmentation culture, and then we can put that in the biotrap and put it in the in situ microcosm unit and make that comparison. All kinds of electron donors, anything that you can imagine that's out there and available commercially, can and probably has been used. The same thing can be said of different electron acceptors. Again, whether you're talking about chlorinated hydrocarbon sites, whether you're talking about hydrocarbon impacted sites, or novel or emerging contaminants. So just to give you a quick example here, this is a case study of amended in situ microcosms. We've got a shallow aquifer here containing TCE, and basically we're seeing a DCE stall. The client's interested in biostimulation as well as bioaugmentation. So the questions that we want to answer are, first of all, are the microorganisms capable of reductive dechlorination of TCE to ethene? Are they present under M&A conditions without biostimulation, without bioaugmentation? Will the addition of electron donors stimulate these organisms? Will a bioaugmentation culture survive in this environment? And is a bioaugmentation culture actually necessary? And on the chemistry side, of course, we're looking for the formation of daughter products and whether the formation of those daughter products are going to be stimulated by either bioaugmentation or biostimulation, the combination of the two. So again, just to remind you, this is the way that the study would be designed, with a in situ microcosms for as a control, a biostimulation unit, and a bioaugmentation unit. So in this particular case, we look at the results of the analysis of the control unit, and we see that dihalococoides is present at a concentration of about 10 to the 2. We're also picking up vinyl chloride reductase. So remember, one of the questions was, is there any reductive dechlorination going on now? And the answer is yes, because remember, those dihalococoides are not going to be found in the biotrap unless they're actively growing and reproducing, and are currently being able to detect, and of course, being able to detect the vinyl chloride reductase also indicates we've got reductive dechlorination going on as well. So I'm going to just bypass some of these things where I'm repeating myself. What about the biostimulation unit? So the biostimulation unit had an electron donor added, and as you can see here, there was definitely an increase in the concentration of actively growing dihalococoides, increases in concentration of vinyl chloride reductase, also detection now of TCE reductase. So one of the questions was, is the electron donor, the biostimulation amendment, going to increase the concentration of dihalococoides? And the answer to that question is certainly yes. And if we look at the chemistry in the M&A unit, you see we're only seeing TCE and DCE, so a classic DCE stall. But in the presence of the biostimulation amendment now, as you can see, been a stimulation and transformation of TCE to DCE, and now we're also picking up vinyl chloride and small amounts of ethene as well. So, sorry, I've pretty much answered those questions. So for sake of time, let me go on. Now, what happens with the bioaugmentation unit? If you look at the bioaugmentation unit, you see very high concentrations of dihalococoides and very high detections of the 2 functional genes as well. One of the questions, remember, was, will the bioaugmentation culture survive? The answer is obviously yes, because we analyzed that biotrap that we loaded the bioaugmentation culture on. We know what the concentration of dihalococoides and the functional genes were in the biotrap before it went out for incubation. We look at it after incubation, and we see that those concentrations are comparable. So does that bioaugmentation culture survive under in situ conditions? The answer is clearly yes. And there's also, as we see, a stimulation in the production. I guess I'm repeating myself. I'm sorry. The TCEA reductase and the vinyl chloride reductase. If we look at the chemistry, we've already seen the M&A and the biostim, and you look at the bioaugmentation, and you say, yes, there's an increase in the amount of vinyl chloride and the amount of ethene, and TCE is now totally gone. So yes, we're getting improved results, but consider these concentrations down in mole fractions, and you wonder if those results are kind of worth the effort. So going back to our questions, will the bioaugmentation culture survive? Yes. Will it be more effective in reductive dechlorination? The answer is yes. But is bioaugmentation necessary? The answer is probably not. And I've reproduced this slide showing you that level that's generally accepted to be their level required for meaningful rates of reductive dechlorination. And you can see we're almost there with just biostimulation. So with more time or additional biostimulation, we could very well be getting useful rates of reductive dechlorination without bioaugmentation. Of course, if we decided to go in that direction, we could use biotraps to continue to monitor the site to see if that was indeed going to be the case. And if not, we might ultimately decide to go with bioaugmentation. Another thing we can do with biotraps is evaluate the actual in situ response to biostimulation or bioaugmentation. And basically that means that we're doing something at the site to stimulate degradation of our contaminant of concern. We're going to use biotraps to monitor the in situ effectiveness of that stimulation in producing the transformations that we desire, as well as determining and what part of the site the stimulation is occurring. And I'll illustrate that with one quick example. This is an ORC injection at a gasoline leaking underground storage tank. Diagram here, you can see in the red box, this is the treatment area and 3 different monitoring wells. The different colors here representing the concentrations of aerobic catabolic functional genes for degradation of BTEX, and you can see those concentrations were quite low prior to the injection of ORC, but 180 days after the injection of ORC, you can see in the treatment area, those concentrations have increased by many orders of magnitude. So was the ORC effective in stimulating hydrocarbon degradation? Yes, it was. If you also look at the microbiology on those biotraps, not just looking at the functional genes, but looking at taxonomic genes, we can see that over time, as you can see, we've gone to almost complete or 100% aerobic degraders as time goes on. Now we go to the last bullet point here, providing irrefutable proof of biodegradation potential of a target compound. And here's where I talk about one of my favorite applications, and that's stable isotope probing. Now, what stable isotope probing is all about is using stable isotope to prove biodegradation under actual in situ conditions. But before I go on, I want to make sure that we understand the difference between 2 different stable isotope techniques. Stable isotope probing is very different from compound-specific isotope analysis. If you've used CSIA, you know that when using CSIA, we're looking at isotopic fractionation that results from differences in rates of breaking bonds involving carbon-12 and carbon-13. And if degradation is going on, we see a slight enrichment in carbon-13. Well, that's very different from stable isotope probing, because on stable isotope probing, we're intentionally adding to our system compounds that are enriched in carbon-13 so that we can use carbon-13 as a tracer. So to use this method, what we would do, let's suppose we were interested in proving that benzene will degrade under actual aquifer conditions at a site, whether that's under natural attenuation conditions or conditions of biostimulation. We want to know that our compound of interest here is being degraded. We would have synthesized benzene using not carbon-12. 99% of all the carbon in the biosphere is carbon-12. So if we enrich with carbon-13, it's very easy for us to trace that carbon-13, as you're going to see. And so we would take that C13 labeled compound and we would sorb that onto a biosat bead and then deploy that in a biotrap. And once it's deployed in the biotrap, indigenous microorganisms are going to start to grow and reproduce in that biosat bead. And if they have the capability of degrading benzene under actual aquifer conditions, they will do so. And as they do so, where does that carbon-13 go? That carbon-13 goes into biomass, and it goes into products of mineralization. Under aerobic conditions, that's CO2. Under anaerobic conditions, it may be CO2 and methane. So to summarize an overview of the biotrap approach, again, C13 compound loaded onto the beads, it goes into the biotrap. It goes into a groundwater monitoring well, and then we analyze those following incubation. Questions we always get is what happens to the material that we load onto the beads. As you can see here, hydrocarbons and very similar compounds adhere very tightly to the activated carbon component of these beads, and in the absence of any biological activity, they basically stick like glue. Other more polar compounds like MTBE and TBA don't stick quite as well, but we will retain somewhere between 70 and 80% of the loaded MTBE and TBA on the beads during long periods of incubation, making them available for degradation of compounds in situ. Now, of course, you've probably seen diagrams like this before. Microorganisms need something to eat, they need something to breathe. That something to eat is an electron donor. Stable isotope probing only works for compounds that are being used as carbon in energy sources. And the electron donor can be oxygen, nitrate, any sort of respiratory metabolism. And we produce more biomass and, of course, energy for growth and the end product CO2. So if we, something didn't work there. Anyway, if we substitute for the electron donor our C13 labeled compound, there it is right there. That C13 gets incorporated into the biomass. It also gets incorporated into carbon dioxide. And if you notice in the lower right-hand corner here, I've shown rapid growth and slow growth. But for rapidly growing microorganisms, you will see that carbon-13 show up in large amounts in the biomass as well as in the CO2. For slow growth, microorganisms will still utilize that C13 compound, but they're using it more for maintenance, just staying alive, as opposed to growing. So you'll see more of the CO2 get incorporated into CO2, more of the carbon-13 incorporated into CO2 than you see incorporated into the biomass. And you'll see some examples of that in a moment. So once the biotraps are retrieved, we do three different types of analyses. Put my little microbe down there. First of all, we know what the concentration of the C13 labeled compound was in the beads when it was deployed. So when it comes back to the laboratory, one of the first things we do is determine how much of that C13 compound remains there. We know in the absence of biological activity, within statistical certainty, it's still going to be there. So if any is missing, it's been degraded. So we know what the concentrations were or how much of the compound was degraded. We know over what time it was degraded, so we can actually estimate a local rate of degradation in the wellbore area where that biotrap was deployed. We also look for the C13 in the inorganic carbon. Here I'm showing CO2, but remember under anaerobic conditions, we can also look for carbon-13 in methane. And we look for carbon-13 in biomass. And what we typically do is look for carbon-13 in the phospholipid fatty acids, which is basically the skin of the microorganisms. And the reason for that is because there is so much phospholipid fatty acid in the microorganisms, it makes detection a lot easier. However, if you also want to know what organisms are doing the degradation, we can also look for C13 incorporation into nucleic acids and identify taxonomic genes, and in some cases, be able to tell you what organisms or what classes of organisms are involved in that degradation process. Now, I'm going to show you a quick case study here, but before I do, we need to be all familiar with this del 13C notation. This is just the way that scientists measure the carbon-13 content of an organic compound, and basically it's a calculation from a measurement of the C13-C12 ratio of a sample compared to that of a sample a standard, and that standard is a mineral PD belemnite, and the units of this del-13C value are per mil. To give you an idea of what kinds of numbers are going to be typical of source material like crude oil, as well as manufactured or isolated products like benzene, TCE, PCE, MTBE, you can see the values on the scale on the bottom there on the x-axis, and you can see their negative numbers. And that's because they all contained less carbon-13 than the standard PD belemnite. So on this scale, if you move from right to left, it's less, that represents less carbon-13 in the molecule. As you move from left to right, that represents more carbon-13 in the molecule. So for CIP results, even though there's some naturally occurring C13 in these materials, when we're looking at the result of a CIP analysis, if the compound is being degraded, the amount of C13 that's incorporated into the biomass, into the CO2, is going to be so great, there is no misrepresenting or misinterpreting the C13 content for natural C13 versus C13 that comes from the tracer material. So let me show you a quick case study. Here we got MTBE TBA impacted aquifer. We've got an air sparging well that you can see with my arrow here. We've got two wells here that are amended with C13 TBA amended biotraps, and then another well up here amended by C13 MTBE amended biotraps. You can see the MTBE and TBA concentrations here, relatively low. And just to quickly give you some results, these are the average del-13C values of phospholipid fatty acids and DIC from these wells. And I've also indicated here how far these wells were from the air sparging well. So look at the closest well, for example, that was a TBA, a well that received C13 TBA. We have huge positive values for the phospholipids, which means huge amounts of carbon-13 have been incorporated into biomass. How did that get there? Only one way it could possibly get there, and that is if the microorganisms present in that environment can and did degrade that TBA under actual aquifer conditions. We also see a natural DIC is about -23. We also see incorporation of carbon-13 into dissolved inorganic carbon too. But notice how this is fairly low compared to these numbers. So this is a situation in which we've got very high growth rates. So a lot of that carbon-13 is going into biomass and less into CO2. Now we go to the second well, which was 15 feet away, the third one 13 feet away. And as you can see, the amount of C13 incorporation is decreasing in the biomass, but it is increasing in the CO2. And why is that? The reason for that is because the oxygen levels are getting low enough now that the microorganisms cannot be growing as rapidly as they were 10 feet from the air sparging well. So basically they're maintaining, they're staying alive. So a lot of that C13 is going into CO2 and lesser amounts are going into phospholipids. Another quick case study here. This is a leaking underground storage tank. The client was interested in just demonstrating that benzene is being biodegraded. Monitoring well 20 is where we deployed the C13 amended biotraps, and the client allowed us to do a little research here. Instead of deploying one biotrap in the well, we deployed 6 biotraps in the well and looked at the distribution of carbon-13 in the different phospholipid fatty acids And the reason for doing this was simply for our own purposes, to look at reproducibility. And what I want you to see is how reproducible these 6 different biotraps were, not only in the relative amounts of the different fatty acids, meaning that the populations are basically the same in all 6 of these biotraps, and also the level of C13 incorporation into these phospholipid fatty acids is consistent. This is just a partial list of many of the compounds that we've used for stable isotope analysis, everything from BTEX components to novel compounds like sulfolanes, dichlorobenzenes, anilines, etc., phenols, dioxane. Anything that can serve as a carbon and energy source can be obtained, enriched with carbon-13, and we can answer that question for you using stable isotope probing. That question being, can this compound be degraded in this aquifer under existing aquifer conditions or under conditions that I might stimulate, and find out if that answer is yes or no. So what else can you do with a biotrap? I'm running short on time here, but one of the things I want to point out, our clients are very inventive when it comes to novel uses of biotraps. Ones I've been most interested in myself is isolating specific degraders. We've had clients that have incorporated lignin, atrazine, different classes of hydrocarbons, plastics, etc., for the purposes of isolating these compounds. One in particular, I'll point out, atrazine. Atrazine amended biosep beads were basically deployed in atrazine impacted soil. Atrazine degraders grew in the beads, and then they were cultured out of the beads. And what the investigator told me here, which was very interesting, is they were able to isolate 40 different isolates, 40 different isolates that can degrade atrazine from the biotrap, 40 more than they were able to isolate from the soil. So again, I'm sorry, I'm getting a little fast here because I'm running out of time. Here's just a little example here. These are crude oil amended biotraps at 5,000 feet in the Gulf of Mexico, isolating hydrocarbon degraders at that depth. So biotraps have evolved, as you've seen, from the simple idea of the biotrap to in situ microcosms to specialized applications like stable isotope probing. So what are the next steps in innovation? The next steps are, first of all, in situ bioreactor, basically using this ability of biosep beads to promote biofilm formation, to create a device that can propagate indigenous degraders in contaminated groundwater. I've got some diagrams here, but again, I'm almost out of time. So I'm going to go a little bit fast here and show you. Here is a toluene impacted site where toluene concentrations were as high as 400 milligrams per liter. Installing these in situ bioreactors in 2 months, increased toluene removal by 87%. We use C13 toluene amended biotraps to demonstrate the stimulation of toluene degradation by these in situ microcosms, these in situ bioreactors. And as you can see, before the installation of the in situ bioreactors, there was no toluene or very little toluene degradation going on. Here we are at 94 days, we're getting huge incorporations of carbon-13 into CO2. And I would like to add that this particular well right here was actually 12 meters from the nearest ISBR. Now, I don't want to leave out the our newest evolution of the biotrap, and that's the MinTrap. The MinTrap is very similar to an in situ microcosm, but instead of a biotrap We have basically packets of site material or even fine sand that can be deployed in particularly for areas where reductive dechlorination treatment is going on, evaluating the abiotic component of the degradation going on at the site. These are developed by Arcadis and marketed by Microbial Insights. Just to very quickly show you, here are some bin traps deployed in background and in an active ERD area of the site where a lot of reductive dechlorination is going on. You can see in the active ERD area, they've turned black. They've turned black due to the formation of iron sulfides. These iron sulfides are basically responsible, these are active minerals responsible for abiotic reduction of chlorinated hydrocarbons, which can be picked up from the chemistry as well as analyses like XRD. And I'm very sorry, I know that I've come right up against our endpoint here. I'm very sorry for that. Please, if you'd like a copy of this presentation, send me an email, send me questions. I'm happy to address any that you have since we don't have much time here, but I'll turn it back to Dora.
All right, thank you much, Carrie, for this presentation. It was really good as always and very educational. So if you have questions, go ahead and enter them into the GoToWebinar panel. We'll do as much as we can today. And then as Carrie said, if there's other questions we don't get to, we'll be happy to follow up with you afterward. So the first question that we have, Carrie, is when you're talking about biotraps, detecting the status of the microorganisms, can you clarify that this is not in real time, but is quantifying the overall microorganisms that are detected within a time period? Is that accurate?
Okay, if I understand the question correctly, we're incubating the biotraps for 30 to 45 days. And so the biofilms that are forming in the biostat beads are in equilibrium with the microorganisms in the surrounding groundwater. And of course, that can change a little bit over time as incubation takes place. But again, two things I'll add to that. One is remember that the microorganisms have to be actively capable of growth and reproduction under in situ conditions to be detected in the biotraps. The second thing is that this also dampens the biotrap dampens some of the fluctuation that occurs during that incubation period. So you're looking at an average biofilm in equilibrium with the surrounding groundwater during that period of incubation. I hope that addresses the question.
I think so. I think, you know, at this point, there's not any type of real-time sensor technology that's going to tell us that you have colonization of certain items or anything during the period. It's just that overall deployment period.
Right.
Okay. Can you talk a little bit about PFAS and if we've used it for biodegradation studies?
I've not been involved in any PFAS studies, and I would refer that to you, Dora, to comment on. Do you know of any applications of yet of biotraps with PFAS?
We do have a current grant study that's being done, and I can't get into the details of it right now because it is currently underway, but is looking at amending biotraps with some PFAS compounds and then looking at, you know, potential, you know, degradation that is occurring in those. So hopefully sometime in the next 6 months to a year, we'll have some results for this that'll be published, and we'll be sure to share that with everyone. But at this point, these are kind of the first studies that are being done to my knowledge. Okay, Carrie, so the ORC injection case study that you did, there was a blip after the injection with little aerobic degraders and a lot of SRBs that were being detected. What do you correlate that to? Do you think it's related to a temporary change in geochemical conditions?
Yes, I do. And it's since that was not our study, that was a client study, I'm not privy to all of the data from that site, but here's my interpretation. My interpretation is that with the initial ORC injection, and again, I don't know the frequency of injection, but with the initial ORC injection, we got a lot of partial degradation of hydrocarbons, and those partially degraded hydrocarbons are excellent carbon and energy sources for sulfate reducing bacteria. So if we had areas of the treated aquifer that basically became anoxic and there was sulfate there, and obviously there was, the sulfate reducers are going to be turned on until the sulfate is depleted, and then oxygen is the oxygen diffusing into that area is going to be the only game in town. So without knowing the exact details of where the ORC was injected and how frequently, I can't be more specific than that. But I have seen this kind of thing before. If oxygen is initially available, but then becomes limiting in an area for some reason, you stimulate a lot of sulfate reduction because of all that partial oxidation of hydrocarbon that's taken place. Those partially oxidized hydrocarbons are excellent substrates for sulfate reducers. But again, the sulfate reduction is then going to go away if the sulfate then becomes limiting.
Awesome. Okay, since we're running out of time, we'll just do one more question, Carrie, but we do have a lot of comments that people say this was a fantastic presentation. I think everybody appreciates your time, so I wanted to share that with you. But the last question is thinking about other ways that we can use the capability of these beads to attach and understand microbes. Can you talk a little bit about being able to do metagenomic sequencing to be able to better assess who's there? Also, have you done studies where you can isolate the organisms directly from the beads? And then can you talk some about the work that we've done using microscopy like SEM or TEM to be able to analyze the biofilms?
Okay, there were three questions there. What was the first one?
About metagenomic sequencing.
Metagenomics, I have not personally used it, but I know that there has been some use there. Dora, would you like to comment on that?
Yeah, I can talk a little bit about, we have a great study that if anybody's interested, you can reach out to me. We've done a presentation, we could do one for your group or share this information, but we used some biotraps and next generation sequencing technologies to look at shifts of microbial populations over time and some statistical analysis to help us understand how those shifts were occurring. And it was a pretty interesting story because we saw some agricultural influence that happened upstream of a sampling event that was unexpected. So we saw some microbial changes that we didn't anticipate that the biotraps helped us to kind of select and identify, plus the, the molecular tools in combination with that really helped us to understand that.
And I know there was one part of that question having to do with isolating microorganisms from the beads. That is extremely easy to do. It is very easy to both load microorganisms onto bioset beads as well as extract microorganisms from bioset beads. Not just the biomarkers, but the live organisms. If you cultivate, it's been my experience personally over many years, anything that I have tried to cultivate with bioset beads in the shake flask or fermenter or whatever it is, these organisms partition into the beads, and you can then separate the beads from the planktonic organisms and continue culturing, and that equilibrium reestablishes itself. So if I look at that from both directions, it's very easy to load anything you want to onto the beads. We've done hydrocarbon degraders, MTBE degraders, TBA degraders, 1,4-dioxane degraders, sulfolane degraders, all kinds of organisms we've cultured and gotten into the beads to put in a biotrap for bioaugmentation or for other uses, as well as then being able to extract or grow out of the beads the microorganisms that form biofilms in the beads. So if I if I took those, for example, those atrazine amended beads after they were incubated in soil, they were basically put in a shake flask with a medium to grow on atrazine. And again, a partitioning took place between the organisms that are forming biofilms inside the beads and the planktonic, or excuse me, the free floating or sessile organisms, planktonic organisms, sorry, then in the culture medium can be easily recovered. There was a third part to that question.
Yes, about scanning electron microscopy that we've done on the beads.
Most of the scanning electron microscopy that was done with the beads was done by me and others early on when we were really very interested in what was going on the inside of the beads. So we've used SEM, we've used live dead stains, and that I guess that's basically all. But we've also, I'm thinking of one other occurrence in which I was growing a thiobacillus, and we were also seeing sulfur crystals that were being formed on the inside of the beads from the metabolism of the organism. But early on in understanding what was going on the inside of the bead, it was very useful, particularly to use SEM. I'm not sure if that's helpful to the question.
Yeah, I think it is. And we also use SEM commonly on the MinTrap samplers that Carrie showed you guys in the presentation today. Those are great ways that we can see the microbial interaction and state changes that are happening inside those. So it's kind of the same concept with the beads, that it could certainly be done if there's interest in that. All right, Carrie, well, thank you again for giving us such an informative presentation and being willing to take your time to do this for us. I really, really appreciate it. I always enjoy hearing you speak. And thank you to everyone in the audience for taking your valuable time to be here and listen to us today. And I know that Carrie and I both want to share information with you. So if you have questions, feel free to reach out to either of us or to send an email to info@microbe.com, and we'll be happy to provide you any information that we can. But thank you, everyone, and thank you very much, Carrie, for your time today.
Thank you.